All right, let's really quickly talk about water sampling and how this is going to be different from the other types of sampling that we've seen so far. So to get us started with water sampling, one thing that we didn't talk about with our soils or with our other samples is what makes a good sampling plan. So the first thing that I want to make sure that we talk about is the fact that when we sample, things should be representative. So they should reflect a specific population that we're looking at. The next thing is that they should be high quality. So they should be accurate and precise. They should be reproducible, which means if we did this again, if we repeated our sampling procedure, and with those samples we did our analysis again, we'd get similar results. That's reproducible. Defensible means that we have valid reasons for our specific sampling plan. And then the last is useful. So the data actually meets objectives. And useful kind of seems like a strange one to throw in here, but that's becoming more and more important as we have more and more information about the world. So good sampling plans should be representative, high quality, reproducible, defensible, and usable. So these are things to think about, especially when we do water, because we're going to start to see with water that it is much more spread out and it's much more fluid, no pun intended. So there's a lot of things to think about with these types of samples. So sampling in water actually tends to be easier than soil. And maybe pause the video if you're watching this and say, well, why? Why might that be? Well, water tends to be more homogeneous than soil, so there's less site-to-site variability between two samples collected within the same vicinity. Water is very dilute compared to soil. So it's more homogeneous in some ways than soil is. We saw that there's lots of different characteristics of soil. Water tends to be very homogeneous. Often it's actually easier to physically collect water samples because it can be done with pumps and hose lines, and we have lots of ways of doing this. So we know volumes of water can be collected from known depths with relative ease, which is kind of nice. And we can also sample with different variable volumes. So sampling with 1 milliliter versus 100 milliliters. So volume is going to start to be important when it comes to water. So it tends to be a little bit easier in water to sample than soil for these particular reasons. So when we do a water sample and we take a water sampling, when we're talking about processing those samples, processing the samples can be more difficult than soil. So actually taking the sample is easier, but processing it for analysis is a little bit more difficult than soils. So the number of microbes tend to be lower in water than they are in soil. And so what that means is normally in soil, we have to dilute the microorganism concentration. In water, we're going to have to concentrate the number of microbes to make sure that we actually get something that's representative. So that's important. So for things that are larger microbes, including bacteria, protozoa, parasites, samples are going to be filtered to trap those organisms on a specific type of filter pad. And for bacteria, we often use a filter that is 0.45 micrometers inside. That's an industry standard for a lot of these methods. So when we extract and we isolate organisms from water, the first thing that we have to do is we have to concentrate the sample. So remember that this is very different from soils. Soils, you normally have to dilute or spread out or shake them off the soil particles. Water, we need to concentrate. And so normally what we're going to do is, hopefully what you've seen maybe in 321L, is using a membrane filter, and we're going to use a vacuum to pull water through a membrane. So what's going to be critical here is the type and the size of the membrane that you're pulling organisms through. So we tend to see that pore size of 0.45 micrometers again, because that is related to bacteria. Then what we do is we take the bacteria that's trapped on the membrane and we place on agar or cellulose with some kind of a media to grow colonies. So we go back to our favorite cultural methods most of the time after we do a filter method. And one of the things that's important when we're doing this technique, and the reason that we do this technique in this way, is that we want to minimize the stress on an organism. So if we're stressing out the organism, it's going to die, right? And that's not a good thing if we're trying to quantify things, or it's going to change, or it's going to do something weird just because it's having trouble. Because of all these external factors. So processing them relatively quickly is important. Making sure the vacuum pressure is at the correct PSI is important, and then making sure our samples don't desiccate or dry out is going to be really important for water. So this is going to be a table that I'm not going to go through in depth in this video, but you can take a look at it. And what this shows you is all of these different types of methods. We're going to stick primarily to the membrane filtration method and some other presence absence, MPN tests and things like that for water, but there are lots and lots of methods out there. So I just want to show this slide to kind of show you that different volumes of water are needed for different tests, and there's different applications of each of these tests. So you may have to do one of these tests in the future if you end up in a job that has a lot of lab work. But if you're somebody who works a little bit more on the consulting side, you may be using just a regular membrane filtration test or just interpreting results from those. So filtration method, I just wanted to show this type of slide. So what you'll do in a filtration method is you'll have a vacuum pump that gets applied to the bottom component. So normally that is an Erlenmeyer flask that has a vacuum attachment to it. And then you have a piece of filter paper, and then you have a funnel on top where the sample is pulled through. And you set up this arrangement with a clamp to hold that top piece and that filter together with the Erlenmeyer flask at the bottom. Then what you'll do is you'll gently wet the top of the filter so that the filter is prepped and ready to go. And you very gently apply the vacuum and then let that sample be pulled through. And you want to pull a known volume through if possible, because you're going to want to know how many organisms per volume you're getting. And then once you're done with that and you take out that piece of filter paper, that piece of filter paper could be dissolved in saline if we wanted to, or it could be put directly on a pad or an agar plate in order to grow colonies of these microorganisms. Filter size wise, again, that 0.45 is going to be important to us, but there's many different sizes. So if we were doing research in the lab and looking at different microorganisms, we might want to start to think about different filter sizes. So this is something that starts to become more and more important if you're looking for very specific organisms. We use that 0.45 micron because we just want to look at bacteria in general. But let's say that we wanted to look at protozoa, or we wanted to look at we wouldn't use this for viruses, but we could use it for other types of organisms, algae, things like that. We'd want to start to consider our sizes. So that's going to be important to us when we do water sampling. And then the other thing that I want to point out here is that the volume of water does matter quite a bit. So what you're going to see is you're going to see sample type here and then the volume, and there's going to be an X in the column if that's the correct volume. So we're going to talk about fecal coliforms in upcoming lectures, but basically these are organisms that are found in feces that cause disease in warm-blooded animals. And so if we're looking for something from a surface water, for example, a surface water, we would use 1 milliliter as our volume when we do detection of fecal coliforms. That's kind of what this table is telling you. So that's important going forward because we're going to see many different methods, and we're going to see standard methods from the EPA, we're going to see standard methods for MPN, we're going to see all these different things. And with water, the volume is going to be pretty critical. So just to show you that that's why this matters to you. This diagram is just going to show you how we could potentially sample and do sample collection for water from wastewater systems, from water systems that are slightly different. So you can start to see how there's a lot of different pumps involved. There's different methods we can plate, we can do assays, we can do all sorts of different things. So this is just to show you that there are different methods out there for analyzing your water samples. The other thing that I want to make sure that we hit is that most of the time we're going to be talking about bacteria in water, but protozoa are going to be a big thing for us in tap water and surface water, especially as environmental engineers. So we actually need quite large volumes of water because remember that protozoa, one of the things that we're worried about is that the oocysts or the cysts associated with these protozoa are actually what we care about, and those are small. So we need to have large volumes of water to detect very low numbers. So that's critical with protozoa. So same idea here. We will try to filter the water through in this case, it's a cartridge, which you can see on the right side of the screen. And then that cartridge itself actually gets sealed and stored on ice and sent to a laboratory that needs to be processed within 75 hours. And extracting the cysts is a very, very specific procedure that's done by a lab where they use fluorescent dyes to provide contrast to be able to detect them by microscopy. So this is not something that can be done instantaneously in the field. We can't detect protozoa as soon as they're in the water. That's just not possible right now. That's something that people are looking to improve in the future. But just to show you that it gets a little bit more complicated when we have specific organisms. And then same thing here. This is going to be the procedure for extracting the protozoa in a little bit more depth. So I'll leave this slide here for you to take a look at. And then the last thing that I want you to think about before you move on to the next pre-lecture is just thinking about designing a water sampling plan. So the scenario here we have is that you need to determine whether or not your private well at home has Giardia lamblia. And then how would you determine if this protozoa is present? So just jot down some notes about how you think you might do this based on what you've learned in this pre-lecture. And then in the next coming pre-lectures, we're going to talk about MPN method and indicator organisms.